Since a lot of you expressed a strong interest in learning more about diabetes, I thought I would do another video where I talk a little about the differences between type 1 and type 2 diabetes and what's kind of the state of the art in terms of where the field is now, thinking about what causes diabetes, how do we treat diabetes, and how in the future might we treat diabetes. So this is going to go deeper into the topics that we learned about before when we were talking about kind of the structure of insulin, as well as the functions of insulin. So we went into all of that crazy ways in which insulin was able to help control our blood sugar regulation. So we'll do a little review about that. And then we'll also go further into what gets disrupted in these various forms of diabetes. So our type 1 diabetes, this is also called insulin-dependent diabetes sometimes. And that's where you have this like autoimmune disorder where your body is actually attacking its pancreatic beta cells, so those cells that actually make the insulin. And then there's type 2 diabetes. Type 2 diabetes is the one that typically has a later in life onset, and your body isn't really responding to that insulin signal. And then later we'll talk about, later we'll talk about how later in the disease, the pancreas doesn't make enough insulin because during that initial stage, it's kind of like making too much insulin and there's a bunch of bad stuff going on that's going to start damaging those beta cells. This is also, type 2 is associated often with obesity. So obesity is a high risk factor. We'll talk about how there's like genetic predispositions and then environmental like kind of triggers and things that make it so that you get the double whammy hit, and then people can develop diabetes. Thankfully, type 2 diabetes can be reversible, sometimes through just dietary and exercise, so lifestyle modifications, as well as a variety of treatments. So we'll talk about treatment strategies. So some of it will be just for your own interest. I will point out when things are just for your own interest versus when things are things that I would like you to know, because I do think that you'll find some of these things interesting when it comes to the treatments. But I don't want you to feel like you need to memorize all of this, just so you have in mind, because you're probably going to hear these terms if you go into med school, or even if you're just watching TV, because type 2 diabetes is greatly on the rise. And so you'll often hear those commercials like, do not take if you're on a sulfonylurea or something. Well, we'll learn what a sulfonylurea is. We'll also learn about some of the other treatment strategies. And so if hopefully you don't develop type 2 diabetes, but it's very, very likely that someone you know will, if not you yourself. So let's start back at the beginning and talk a little bit about big picture, what's our insulin doing? So remember that insulin is going to be released in response to our high blood sugar. It's released by the beta cells of our pancreas, and it's going to basically tell your muscle cells and your heart cells and your adipose tissue, so your fat cells, it's going to actually put those glucose transporters to the surface. So those cells are then going to basically take their preformed insulin or the preformed glucose transporters in these little vesicle type things, and then it's going to actually merge those with the membrane. So your muscles and your fat cells, they're going to quickly take in the glucose, and then your liver is also going to take in a bunch of, and so then when there's high glucose in the blood, the insulin is going to help quickly bring things back down. So remember, we looked at those graphs and we saw how when those participants ate a meal, their glucose spiked and their insulin spiked. As their insulin spiked, then their glucose started going back down. And as their insulin spiked, well, then we got this decrease in glucagon. So glucagon, remember, was like the opposite of our insulin. And so if insulin was saying, better let some glucose in, glucagon is saying, all the glucose is gone, and it's going to be released by the alpha cells of your pancreas. It's going to counter the effects of the insulin. So when your insulin, when that insulin doesn't just make it so that your cells take in more glucose and lower the blood sugar back to normal, it also makes it so that they do stuff with the glucose. So it promotes glycogenesis. So basically linking up those glucoses into glycogen. We'll also see later in the semester how it does things like promote lipid making. So in your adipose tissues, your fat cells, when they take in the sugar, So they take in more of it in response to the insulin signal. And then they're going to basically link it up into to form fatty acids and then triacylglycerides. So they're going to store that as sugar as fat. And so that stuff is going to be stimulated by your insulin, whereas your glucagon is going to do the opposite. So remember that your liver has the receptor for the glucagon. So what your liver is going to do is the glucagon is going to signal for it to break down glycogen and ship out glucose. So your liver is going to start shipping out glucose in response to glucagon. But insulin is going to suppress the secretion of the glucagon. And so that's what's going to happen then is that you won't get the glucose being released by the liver when you want the liver taking it in. So insulin is able to have these effects. And now we're saying with diabetes, either we're not making enough insulin, which would be the case with type 1 diabetes, or our cells aren't responding adequately to the insulin. So we're like desensitized, which is the case with type 2 diabetes. If we imagine that we don't have enough of this insulin, what's going to happen? Well, what's going to happen is kind of the glucagon type side of things are going to predominate. Not just because we don't have the insulin action, but we also don't have, like during direct action, but we don't have insulin opposing the glucagon release. And so what's going to happen is we're going to have basically more glucagon, less insulin, and we're going to be really promoting that glucose breakdown and release from your liver. So what you're doing is you're kind of actually shipping out glucose when you have high glucose in your bloodstream. So it sounds really weird, but your glucose would be high in your bloodstream. So we call that hyperglycemia when you have too much glucose in your bloodstream. And so that's happening because your cells aren't taking in the glucose. Because the cells aren't taking in the glucose, inside of the cells, the concentration of glucose is going to be low. Therefore, the cells are like, hey, we don't have enough glucose. Let's make some glucose. Let's free up some glucose. So your liver is going to start making this glucose to ship out. We'll talk later about like lipids and lipid metabolism, but your fat cells, they're going to be like, oh, we need, and your liver as well. Basically, they're going to say like, we need to ship out some we need to break down our triacylglycerides, our fat cells, and ship out lipids to the body. And so now you're going to have lipids building up in your bloodstream, and you have lipids build up in your tissues and things like this. And this can cause all sorts of problems. So it's not just directly having hyperglycemia that's bad, having that high blood sugar, but we get all of these metabolic consequences as a result. Later in the semester too, we'll talk about this condition called diabetic ketoacidosis. Basically what happens is that your cells, remember, they don't have enough glucose inside of them. And if they don't have enough glucose inside of them, they can't do the TCA, the citric acid cycle. And that's where we're going to get the NADH, the bulk of our NADH. So remember, with glycolysis, we could get some ATP, and we could get one NADH, but then, or two NADH. But then the citric acid cycle is where we would get that really big payout of NADH and FADH2. And that, remember, gives us the electrons we need for our electron transport chain, for our oxidative phosphorylation, for the production of ATP. So our cells, if they don't have enough glucose, they can't do this. If they can't do this, what's going to happen is that you're going to get the buildup of this acetyl-CoA. And the acetyl-CoA, we'll talk way more about this, so don't worry about it for now, but it can get transformed into this form, what we call these ketone bodies. And these ketone bodies can get sent through the bloodstream, and they're carboxylic acids. So what they're going to happen is your blood's going to acidify, and this is called ketoacidosis. Again, we'll get into this later in the semester. Don't worry about it now. But that is another connection we'll see with diabetes is we can get this lowering of the bloodstream, this ketoacidosis. And we'll talk a lot more about that. So something to look forward to discussion-wise. So remember, our insulin is released in response to high glucose. That insulin is going to then bind to insulin receptors on the surface of cells. We saw how the insulin receptors could act through like our IRS1 pathways. So these insulin receptor substrates, the insulin receptor, it binds to insulin, it gets activated, it phosphorylates itself, it phosphorylates IRS1. Phosphorylated IRS1 associates with PI3K. PI3K is then able to basically downstream activate PKB, which remember is called AKT as well. What's another term for it? That's going to activate GSK3, which is going to inactivate our glycogen synthase. It's also going to do all other sorts of stuff in order to promote our glycogenesis, so our making of the glycogen, and inhibit glycogenolysis, so inhibit the breakdown. Insulin can also act to basically move those glucose transporters in our muscle cells and in our fat cells and heart cells to the surface so that you could let more glucose in when you have high blood sugar. But how is the insulin itself released? We say insulin is released in response to high blood sugar, but how is it actually released? Well, it's kind of an indirect thing. The high glucose, when it gets into the cells, now what's going to happen is it's going to go through glycolysis. It's going to go through the citric acid cycle, oxidative phosphorylation. We make ATP. When we make ATP, the intracellular ATP concentrations are going to rise. Those are going to bind to ATP-gated ion channels for potassium. Remember that the concentration of potassium inside of our cells is going to be higher than the concentration out of our cells. Remember that special K. Our cells are special. They've got a lot of that potassium. So the potassium would be kind of like diffusion-wise, it would want to go out. It can't normally go out because the plasma membrane is nonpolar and it's this charged molecule, but it can get through facilitated diffusion, and then it would go out of the cell. If that channel is open, the potassium is going to flow out. When the potassium flows out, well, it's taking the positive charge with it. It's making it more negative inside. So we're like hyperpolarizing things. But what happens if that channel is closed is that then we would do the opposite. The membrane would depolarize. It's a little confusing because when we talk about ligand-gated channels, it sounds like, oh yeah, so basically the ligand binds and the channel opens. In this case, however, when the potassium binds, the channel actually closes. And so when the channel closes, well, then you're going to get that depolarization. That depolarization is going to actually trigger a voltage-gated calcium channel is going to be like, oh, the membrane potential changed. It's going to let in calcium. Increases in calcium are then going to make it so that these little secretory granules, so these little kind of bubbles with insulin ready to go, are going to be merged with the membrane, and you get insulin secretion. This is like you get this like first phase insulin response. When you first eat that glucose, you get that quick ATP rise. And then what's going to happen is these pre-made insulin is going to get shipped out. So that gives you that initial like high spike that we saw when we looked at like this graph. That high spike in your insulin is going to be coming from that like pre-made stuff that just needs to get inserted into the membrane. Then, however, you get this kind of like you need more sustained release of the insulin over time to bring your blood sugar back to normal. And so your cells can make more insulin as needed, and you get this steady decrease in the amount of glucose in your bloodstream, and then your insulin levels can go back down to normal. All of this, however, is assuming that your cells are able to make insulin, that you have beta cells in the beginning, that they're able to make insulin, and that the cells on the other end are able to respond to insulin. So with type 2 diabetes, there's various reasons we could think about why those cells might not be able to respond to. We'll go into some of these, but we can already kind of think about the various places that we could get problems. So we talked about the various pathways. Not only do we have this kind of like PKB signaling pathway, but we talked before about our RAS signaling pathway, how we had that RAS-RAF-MEK pathway that was going to then promote the transcription of various genes. All of these players are places where things can go wrong. When it comes to diabetes, it's typically like not just one thing going wrong, and we're not even exactly sure what exactly is going wrong. Both type 1 and type 2 diabetes are usually polygenic. This means there's multiple genes involved. It's not just one. So people often have like genetic predispositions and then environmental triggers. So they have some genes, some versions or like polymorphisms, some alleles of genes, some versions of them that make them more susceptible to getting diabetes. But then if there's some sort of like external trigger, maybe there's weight gain in the case of type 2 diabetes, maybe there's some sort of virus infection, maybe that triggers the autoimmune response, various other kind of environmental factors that can come into play to make it so that even if people are like identical twins, they might have a really high likelihood of both getting the disease. If one has it, then the other has a high likelihood of getting it and that sort of thing, but it's not absolute because there are environmental factors. Knowing all of that, let's go back and let's look a little more about what the current understanding of these diseases is. So we'll talk a little about the difference between our type 1 and our type 2 diabetes. So in type 1, the pancreas doesn't make enough insulin due to autoimmune destruction of the pancreatic beta cells. So there are some times when it's like a single mutation is known to cause the disease, but often it's going to be like polygenic. Sometimes the risk factors in a lot of the risk factors that have been found gene-wise include things like various immune genes, things like HLA types and things that we're not going to get into. But it's this sort of like autoimmune thing. Your body, your immune system is recognizing your pancreatic cells as foreign and destroying them. And so they find like in that up to like 90% or so of a person's pancreatic beta cells can be destroyed in the case of type 1 diabetes. Whereas in type 2 diabetes, initially the body is making enough insulin. In fact, it starts making too much insulin because the body isn't responding to the insulin. And when it's sending out too much insulin and the blood sugar is really high and all of this various stuff, it can cause damage to that pancreas. And then the pancreas is going to start not making enough insulin. And so then you get more like a type 1 situation where your body's not making enough insulin. And so we'll talk more about what kind of ideas are at play about how that destruction or that dysfunction might be coming about and various strategies for treating it. Type 1 diabetes is kind of easier to think about. It's simpler. You've got your pancreas isn't making enough insulin. Well, what you're going to do about that, you just give them insulin. So we talked in the previous video about how their insulin, we have this peptide, so it's held in these secretory vesicles and then released. There are artificial versions of insulin. So basically we can make them in a lab and then we can the patients can inject them. So we talked about how the patients measure their blood sugar, the in the moment blood sugar with those glucometers. And then if it's low, what they can do is if their blood sugar is low, because maybe they overdid it on their insulin, then they can eat some glucose. And if if blood sugar is high, which is more common with uncontrolled diabetes, then what's going to happen is you would want to inject insulin. And how much insulin you inject is going to depend on how high your blood sugar is. And so there are like the monitors often have ways where they calculate how much you need. A lot of people have like continuous monitors where they basically measures the glucose in your bloodstream all the time and then corrects it by injecting insulin as needed. So the pumps today, they basically measure your blood sugar and they pump insulin when needed. It's important though that the people still make sure that it's really working correctly and that it doesn't overpump and then their blood sugar will be too low, so hypoglycemia. It's not perfect. Pumps of the future are hopefully going to also give out glucagon. This would be kind of like an artificial pancreas, they call it. Basically, you would have some sort of continuous monitoring system. It's attached to your bloodstream. It's continuously monitoring your glucose levels, giving you insulin when you need it, and giving you glucagon when you need it is the future hope. But now they just give insulin. There's been problems with trying to deliver the glucagon, but that is kind of like the hope of the future, is to have this complete kind of artificial pancreas. Another hope of the future is basically there was initially all this like hype about, oh, we can just transplant the islet cells, but then people had to like those cells that produce the insulin. Why don't we just transplant them from someone that has them? But then the patients have to be on like immunosuppressants their life, and that's not good. There's the hope is that they can like now that there's all this stuff with like induced pluripotent stem cells, where maybe you can take cells from a different part of the patient's body and convert them into beta-like cells that can then produce insulin. And then the person wouldn't be like reacting to that like it was foreign because it's themselves. And then they can basically would be able to produce insulin. Another problem with kind of like the transplant strategy is that those cells are really, really sensitive and they need a lot of nutrients and oxygen. So often, like when they have transplanted them, they've actually been into the liver, like portal system and things where they would have more oxygen. That stuff is not stuff that you're responsible for. That stuff is just cool stuff. But let's talk a little bit more about type 2 diabetes now. Now, type 2 diabetes. Here, initially, the body doesn't respond strong enough to insulin because the cells have become desensitized to it. We'll talk more in a minute. And then in later cell stages, the pancreas doesn't make enough insulin due to destruction and or dysfunction of the beta cells. So this paper, they're basically showing they had people with diabetes, and they kind of took these people with diabetes and they looked at their fasting plasma glucose. So if they're just fasting, what's their glucose levels? You can imagine that people earlier in the disease, they would have a lower fasting plasma glucose. People later in the disease, they would have a higher fasting plasma glucose. As the disease gets progressively worse, then the amount of glucose that's going to be, the like degree of hyperglycemia is going to be worse. In these cases, so you can imagine kind of this over a course of time rather than just plasma glucose levels, that they measured as plasma glucose levels because they're measuring different people, not the same person over time. But you can think of this as kind of like a curve over time. And you see that you have this inverted U. What this is saying is that in the beginning, the patients, this is measuring the insulin. So as these patients in the beginning stages of the disease, the amount of insulin they produce actually increases to really high levels. It has to do this because your pancreas is sending out the message, but the cells aren't responding to it. And if the cells aren't responding to it, it's like the pancreas is like, hey, don't you hear me? And so because you're not getting that counter response, remember we have this like feedback. So the insulin gets shipped out, the glucose gets taken in, and then because the stimulus for making more insulin is relieved, the insulin stops getting shipped out. If the cells aren't taking in the glucose in response to the insulin, or they're taking in the glucose, but they're not doing their other stuff that then produces other signals that tell the pancreas, hey, we don't need to do this anymore, then what's going to happen is that the insulin is going to keep getting released. You're not going to get that feedback inhibition. The insulin is going to keep getting released. And so you end up having these high levels of insulin. But then you see later in the disease, as things get even worse, well, now your insulin levels actually fall. And so you actually start not producing enough insulin, especially to keep up with the decreased sensitivity of the cells. So let's talk a little about theories for why this is. This is kind of just more for your own knowledge. I would like you to know about that like inverted U, where initially you start, you make too much insulin because your cells are responding, and then you don't make enough insulin because your pancreatic cells become damaged. This part is going to be kind of just further information because it might be of interest for you. Why do we get this desensitization? And why do we get this decrease in the beta cell production? Let's start with the desensitization. There are a variety of theories for this, and it's probably more multifactorial, so multiple things are contributing. One of the ideas is kind of lipid toxicity. So what happens is that when makes the connection between like obesity and diabetes, or one of the connections. If you have too much fat, basically your adipocytes, they're going to send out fatty acids. The fatty acids are then going to go to your muscles and to your liver, and then they're going to build up in your muscles and your liver. That's going to cause a bunch of problems, and you're going to get this like inflammatory responses and bad things. all of this is going to then make it so that these cells can't respond as well to the insulin. Another thing is that you could have basically any sort of dysregulation at any of those downstream steps, whether it's our AKT signaling, our RAS signaling, and the transport of the glucose. So all of those different things are different ways, different places that can kind of be damaged and therefore cause this desensitization. When you have this desensitization, well, now remember that you're going to be sending out a lot of insulin, and you're going to have high blood sugar as well as high lipid levels because, well, if you have that like glucagon signaling, your cells, remember, they're going to be sending out lipids, and those lipids are then going to be causing further issues. So all of this can cause this destruction or dysregulation of your pancreatic beta cells. This can then, basically, one of the things that is bad about this is amyloidosis. So amylin is this peptide that's co-secreted with insulin. It's actually stored in vesicles along with insulin. And so when you release insulin, you also release some of this amylin or amylin. I don't know. I've never really heard it said, but it is important. It's about like a 15-to-1 ratio. So you release about 15 times the amount of amylin as you release insulin. But if you're releasing a ton of insulin, you're also going to be releasing a ton of amylin. And you're going to remember, because you kind of store that insulin in those vesicles, if you're co-storing that amylin, what can happen is it can actually polymerize into these fibrils. So these long chains that can then form these clumpy things called amyloids. That can then build up and destroy the pancreatic beta cells, or if not fully destroy them, at least damage them. So you might think that this amylin is all bad, but amylin is actually good in some cases too. So it's actually a treatment strategy for in the later stages of type 2 diabetes, because what amylin does is basically it inhibits the release of glucagon, so you don't get that continuous signaling to like break down glycogen, to break down our triacylglycerides, to send that for the liver to send out the glucose, to send out the fatty acids and all that stuff. And so you don't get the glucagon release. So this is going to be inhibiting the glucagon release. So that's good. And it decreases the food intake. So that's good. It does a variety of other things, such as decreasing gastric emptying. So when you eat carbs, they don't get shipped out of into your bloodstream as quickly. So it's like more gradual. So if you don't have that first phase response, we've got that insulin kind of ready to go and is able to respond well, then you still have the potential for, like, if you give it time, things can get to normal. So there are good things with amylin, but you don't want a ton of it building up and damaging the cells. So amylin was one of the strategies that we could actually use to treat diabetes. Now let's talk about some of the other strategies. That's like, the amylin's kind of like a newer, weirder one, but there are simpler things that you can think about. The first is just diet modification and exercise. So remember that one of the theories was like that you have the lipid buildup, this triacylglycerides, that fat toxicity that's causing the inflammation and all sorts of problems. And so that could be, if you then increase your exercise, decrease, like modify the diet, there's various things that you can do in order to kind of keep your blood sugar under control and prevent the formation of full diabetes if you're like in a pre-diabetic stage where you're kind of like sensitive to the insulin, desensitized to the insulin, but you're still making insulin. So that's where, like, those initial change times, we can kind of reverse what's going on, hopefully. But say things go further. Now we need to address things with medications. The kind of like areas that we can look at are in the production or release of insulin. So some type 2 diabetics, it's typically further on in the disease progression, they might actually need to inject insulin because, remember, their cells will stop making enough. They can also use things that actually promote the secretion of the insulin. We'll talk about this in a second. They can also have things that affect how the body responds to the insulin. So these would be what we call like insulin sensitizers, as well as like the absorption of carbs from the GI tract. So kind of like, and slowing it down. So we talked a little bit about how amylin can help with that. There's also this like alpha glucosidase inhibitor, which basically it makes it so that you can't break apart like the carbs as quickly in order to, you can't like free up the glucose. And then the renal absorption of glucose. So basically, we talked about like the facilitated transporters of glucose, those like LET transporters. There's also the sodium glucose transporter 2 or SGL2, which is in your kidney. And basically, it's like a sodium glucose transporter. So it's one of the co-transporters. And what it does is basically when you're building up this glucose in your blood, that blood is then going to reach the kidneys. The kidneys is going to be able to take up some of the blood and like reabsorb it, and it's going to put some of it is going to end up in the urine. You have excess glucose in your urine when you have uncontrolled diabetes, when you have this like hyperglycemia. And the idea is that, I mean, like normally you would think that that would be bad. Like it's a bad sign when you have that. But if we can actually promote more of it being urinated out as opposed to just staying in the blood, then that would help lower the blood glucose. And so by inhibiting the kind of like reabsorption, so inhibiting the kidneys from actually taking the glucose back in, then what you can do is you can have more of it be peed out. So that's one strategy. We talked about these strategies. I'm not really going to go into the sensitivity or these insulin sensitizers, but you can imagine that if the cells are desensitized to insulin, if you could do things that makes the cells more sensitive, that'd be good. These typically have like complicated mechanisms of action, which aren't really fully understood, but they're commonly used. It's like metformin, this TCZ are what they're typically called. Incretins, basically, there are these other hormones that are secreted by your gut. And kind of like similarly to amylin, they kind of promote the action of insulin and they cause the insulin to be more effective, various things like that. And so if we could actually just provide these hormones themselves rather than some sort of medication, these things that the body naturally makes, let's just add those incretins in directly that would normally be released by our gut. And so you might hear this like incretin effect or incretin hypothesis or something like this. Basically, you get different effects on your glucose levels, whether you ingest things or whether they're like injected into you. And some of this has to do with the secretion of these incretins. Again, like with this stuff, this is not stuff you're responsible for. Just know kind of like the main different places that you could affect. So the production release of insulin, the sensitivity to the insulin, and then don't worry as much about the absorption in these factors, but just the idea that you can kind of address the root cause or you can redress what's down the stream from it. So let's talk a little about these insulin secretagogues or whatever. Basically, they make it so that insulin is secreted more. The one I want you to know is sulfonylureas. So let's actually look again at kind of the idea about how the insulin is released through these channels. So glucose comes in, ATP levels rise. When ATP levels rise, they close that ATP-gated calcium channel or potassium channel. When that happens, basically you get this depolarization because you're not shipping out the positive charge, the inside stops getting more negative. So it kind of like evens out with the outside, this depolarization. This is going to cause a voltage-gated calcium channel to let in calcium. So remember, calcium inside our cells is normally really low, but this calcium is then going to get released from intercellular stores and voltage-gated channels. What's going to happen now is that the calcium is going to cause the release of this kind of merging of these insulin secretory vesicles with the membrane, let out insulin. But if we inhibit the channel, well, now, like if we kind of mimic the effects of ATP, close that channel, we'd be able to release the insulin even when there wasn't glucose around or when we didn't have those high ATP levels. If you look at the names of kind of this channel subunits, so it's got these like, it's a tetramer. It's a 2 tetramer basically. It's got these tetramer of these subunits, these Kerr subunits, which is where the potassium goes through. And then it has these SIR subunits around it. The SIR stands for sulfonylurea, and because this is actually where the sulfonylureas bind. So this like tolbutamide, that's one of these sulfonylureas. And so when it binds, it causes the closure of the channel. So it's doing the same sort of thing as our ATP. It's closing the channel. When we close the channel, we promote the opening of the calcium channel. The calcium channel, then we get like all the calcium signaling called modulin, all those good proteins that are then going to make it So the secretory vesicles will merge with the membrane and let out the insulin. So various treatment strategies, just again, this is like not stuff that I need you to memorize or anything like that. I just know that a lot of you are interested in it and it'll come about. But I would like you to know that kind of like the main differences between type 1 and type 2 diabetes, the inverted U of type 2 diabetes, why insulin is so important, and just kind of hope that you found this interesting.